Assalamu alaikum dear students, welcome to the first lecture of the cell culture and technology. So today is the introductory lecture and we will learn about the basis of the cell culture, introduction and history of the cell culture, also the scope of the cell culture. The topic will also include the advancements in the cell culture technology. So today is the basic lecture, and we will also discuss that what is going on in the current era of cell culture and technology. So we are going to start from the introduction, that what is cell culture. Cell culture, we can define it, that is the process by which the animal and plant cells can be grown under the controlled environmental conditions. In routine practice, it referred to the culturing of the cells derived from the plant or the animal cells. The advancements, if just this is the first slide, if you can see here, this is the definition again, that the ability to grow the cell culture, i.e. in the controlled environment. In controlled environment, we can say that in the laboratory. So it is essential usually for the biotechnology and the research. So if we were discussing that what is its scope. In the current era, this is we can say that this current era is called the era of biomedical sciences, or we can say that is the era of the cell culture technology. So what is going on in this era, that the cell culture technology is playing a major role in the biomedical sciences research. Previously, we were used to study this cell culture for studying the basics of the cell biology and interactions between the diseases and their causing agents. But now we can say that many drugs and the cell processes, we can give the example like the cell processes in the nutritional studies, the cell processes in the drug development, the cell processes in the vaccine development, the cell processes in immune histochemistry studies, and many cytotoxicity studies of the natural and chemical products, also the cell viability studies, maybe the cell viability, especially in some eras like in the cryopreservation studies, we check the cell viability. Also the studying the effects of the new drugs. And another area that is a very big area, that is the cancer research. Cancer research, usually the cell culture is playing a major role in the cancer research studies, and we use many cells when we are doing the cancer research. Next there will be slide in which I will just show you some cell lines which we use to study in the cancer research studies. Also the study of various the functions of various chemical viruses, also radiation effects, also the drug effects, the peptide effects, the protein testing, many enzymes linked, and many other studies. We use the tissue cell culture. So then another here is question arises, then if we were talking about the cell culture, so then what is the tissue culture? And tissue, I previously told you in the classes that many cells form a tissue. Cluster of cells is also called a tissue. So in tissue culture, what usually we do, that the tissue culture is the term used for the process of growing cell artificially in the laboratory. So sometime we talk about the cell culture, sometime we say the tissue culture. So basically tissue is when we are growing the cells at like in the mass collection, and we do the cytotoxicity studies. So then this is not only the single cell, but we are growing the tissue now under some specific conditions in the laboratory. So we are referring the term tissue culture here. So tissue culture involve basically both the plant and the animal tissues. We sometimes derive the cells from the plant tissue, we sometimes derive the cell from the animal tissue. So we do both type of testing in cell and tissue culture technology. So tissue culture produces the clone in which the all products cells have the same genotypes unless affected by the mutation during the culture. So this is when we do some experiment in which this type of changes occur. So in introducing the cell and tissue culture, there is another slide that we can say that the cell culture can be defined as the process of cultivating the cells and the tissue outside the body and of an organism. Usually the term in vitro basically means inside the test tube. When we do some experiment outside the body of the organism, we refer the term in vitro. Same other term in the in vivo when we do the experiment or the animal studies work inside the animal body, so we term as the term in vivo. So this is the term in vivo and in vitro. So don't confuse between these two terms when you read. In an artificial environment, usually when we doing the cell culture, the in vivo condition, what we do that we provide the artificial conditions like the in vitro. And how we do this, that we maintain the temperature, we provide them the nutrition, we use the antibiotics for protection from the microorganism. So this is a created environment usually, that's why we use the so this is another slide. You can see that how the plant and animal tissue is collected, and then we grow the primary cell culture, and then is the subculture, and then we go to the cryopreservation of the cells. Here we sometimes use the term copying of the cell culture, that we use the copies of the cells and store them, and we can store them for the long term in the cryopreservations and cryoprotection, usually using the standard cryopreservatives here for the cryopreservation of the cells. And so in the cryopreservation of the cell, usually we use the liquid nitrogen where the temperature is minus 196 degrees Celsius, and we maintain them for the years. These cells and tissue culture, we maintain them in this condition for the years. Usually the scientists nowadays are also thinking about using of the cryopreservatives, that we can use them at the refrigeration temperature, and sometime maintaining in the laboratory fridge or the refrigerator or the deep freezer, where we can we don't want to achieve at minus 196, but these type of studies are still in process, many working and many not working. So they are doing it. So how the initiation of, there is another slide you can see, and that is slide is telling you how we can initiate the cell culture. So we can the cell culture and tissue culture is derived from the plant and the animal tissue, and then again the primary cell culture and the primary cell culture, and then the growing of these cells in the subculture, and then storage of these cells under the cryoprotected environment, or we can say that under the minus temperature where the temperature is sub-zero. So this phenomena is also called the cryopreservation of the cell culture. So we were talking about the scope of the study. So what is the commercial application? Sometime the student again become very confused that why we do this type of practices. Okay, so the commercial application nowadays, the first commercial application is pharmaceutics. Just I am giving the example of the pharmaceutics, in which we use the antibiotics, and antibiotic testing is all done by the cryopreservation. Nowadays experiments are going on the probiotics and probiotics generation. So where also we test these all probiotics and all these processes of development of probiotics using these cell culture techniques. Then another is the human protein, like insulin. This is another big area of the research where we can talk about the scope, like these diabetic, the studies on the diabetes. Largely the insulin studies and the insulin resistance studies, also the studies about the development of the type 2 and the type 1 of diabetes. This also involve the cell culture research, also the enzyme research, complex molecules, steroids research. Yes, these all type of researches usually we use the cell culture to study the also the one big area, another big area is the food storage. This is also we use for the different especially food storage and cryopreservation of food and other processing of food, and we use. So this is another slide for the history. You can see that how these cell culture studies develops. This cell culture studies develop, you can see in this slide that this process is started from 1911. 19th century is the usually we can say that this century is started in the 19th century, and then in 1920 you can say that the composition of the solutions were formulated. Then 1940s the antibiotics were firstly used in cell culture, so they introduced the antibiotics, and then in the 1960s the carbon dioxide incubators become widely available. So this was a breakthrough. Usually this 1960s carbon dioxide incubators, so the incubation become possible here. We can say that not only the incubation, the growth become possible here, and we were able to produce the cells in the bulk. So whenever this when we do in the laboratory, what we do first, that we go in hurry for copying the cells and storage of cells and use of the cells for the next so this was like sometime about the history. Ross Harrison, it a big name in 1907. That cell culture, we can say that he is a actual the game changer. You can remember that name. Also another, basically it was in 1885, 15 years and 20 years before Rouse, that the Rouse was the scientist in 1885 who previously used, I think, which I remember, he used the embryonic chick embryos. Yes, embryonic chick, I remember that he used the embryonic chick cell culture, and this was the first cell culture experiment which I so this is the another terminology. Previously I told you that we use the terminology cell culture and then the tissue culture. So you can now understand the cell culture and tissue culture difference. That the cell culture is the general term in which we use for the you can say the growing of the cells in the artificial environment, and when we grow many cells in the artificial environment, we give them term the tissue culture, and then the organ culture. So this is usually a new era. The people are going to cryopreserve whole organ, and the research is going on the whole organ cryopreservation. Same the whole organ tissue culture, especially in the bone growth. You can many research paper you will find in that area of organ cell culture. So don't confuse that this is something very new. So many things are going on. So the animal cell culture, this is on the organ cell culture is also a part of the so this is again a slide of application of the cell culture to study the normal physiology, biology and biochemistry of the cells, and then the effects of the drug, radiation, and the I previously told these all things to you. Also the if something we have used that the used for the gene transfer, this is new in that slide. You can say that the large scale manufacturing of biological compounds, and especially this cell culture is very common in the vaccine production, insulin, interferons, probiotics, these all type of things I previously discussed with you. So again talking of the basic cell culture, basic cell culture, previously we discussed this all introductions, that what is the cell culture, and this is the cell culture is the used for the previously we were talking that this is their use, and animal and tissue, animal cell and tissue, even organ can be cultured like plant cell, tissue and organ. There is too much research you can nowadays they are practicing the organ culture research, whole organ culture, whole organ cryopreservation. Many things are going on in the usually what we have done every time we talk that it is the era of the cell culture research, era of the biomedical sciences. So we have done basically many things. The scientists have done many things and they are doing a lot. So what previously, just these are the glimpse that I am telling you now, that what we have previously done. So we have what we have done, that we have discovered the antibiotics, we have discovered the this carbon dioxide incubator to grow the cells, then the use of trypsin, this was also a milestone. And the use of trypsin, basically what we do, why we use the trypsin. Usually the trypsin is used to remove the adherent cell to subculture to further growth. Usually when we transfer the cells to the other culture vessels, when we usually transfer in the subculture to the more petri dishes, more flasks, more vessels, culture vessels. So we use the trypsin to remove the adherent cells from the subculture. Also the optimization of the chemically defined culture medium. Nowadays many things are many researchers are going on to optimize the different chemically constructed medias. They are going for the natural products. Many scientists are optimizing the medias using the natural products. So then the probiotics, we are using the probiotics nowadays. embryo transfer technique. So it is boom, yeah, of the research, that the embryo transfer technique is now, we can say, the test tube babies are nowadays every time, everywhere you can see these embryo transfer clinics. In vitro fertilization, especially in animals, many good breeds have been developed using in vitro fertilization. The tissue cryopreservation and transplantation, I previously told you that now people are going for the whole organ cryopreservation. So what the condition we usually need, this is slide you can see that these condition usually we need in order to grow the cells. We know the condition must be just right for each cell type. The cytologist must therefore consider the following things, like the growth medium, the type of the growth container or fermenter, and these all things in previous slide you have seen. So what is the difference between the plant and the animal cell culture? Animal cell line have limited number of cell cycles before they begin to degrade, while other difference is the animal cells need frequent subculturing to remain viable. Subculturing is the basic thing. Usually when we do the subculture, then we then we cryopreserve and copy the cell, and again we grow the cell, again we subculture, again we cryopreserve the cell. So this cycle usually continue. And then the tissue culture media is not fully defined as that of the plants. In addition, that inorganic salts, energy sources, amino acid, vitamins, etc., they required to the addition of the serum, like bovine serum is very, fetal bovine serum, FBS, everybody knows. So this is very common. I previously told you that optimization of chemically defined culture medium is another area of research nowadays, because I have seen in many labs that usually people are going to optimize the new media. Some people use the natural product, some people use the plant extract, some people use the proteins, some people use the peptides, some people are trying the probiotics nowadays. So this optimization is still going on, and this optimization is we can say that this is the new things basically. So and that that is the another slide, that what do cells need to grow. Usually this is the basic lecture, so just these things will make an idea for you people, that that in the cell culture technology, in the cell culture laboratory, what are the common things we use. These are the cell these are this slide was again this was you can say that these basic necessities when we do the cell culture research. So talking about the scope again, that the virology research, I previously told you that the vaccine production is another area where we can use the study of the infection cycles, also the cancer research. These all the scope slide, and then the genetic engineer. So why we do the cell culture? So this was the slide about. So these necessities include the substrate or liquid, the cell culture flask or the scaffold material, chemically modified plastics or coated with the ECM proteins, suspension culture, also the nutrients that include the culture media, also the environment, the carbon dioxide incubator, which having the 37 degree centigrade of the humidity, oxygen tension maintained at atmosphere but can be varied, also the sterile conditions. All conditions must be sterile to avoid the contamination. The contamination is a big issue doing the cell culture research. So what is the primary culture? Primary culture, these usually derive directly from the animal tissue embryos or the adult or normal or neoplastic, maybe the pathological conditions. So the culture either at the tissue, that we can say that we can culture that as a tissue, we can culture it as a single cell if we are very interested in cytology, and then we can say that in vitro, that having a finite lifespan. Cycles, I previously discussed this, that we usually cycle this by starting the cell culture and then growing and then preservation and then growing and then subculture and then preservation. Also the differentiated phenotype they retain. So these are this is another slide, how we usually isolate the cell line for in vitro culture. This tissue, and then the cell or the tissue culture in vitro, and then the primary cell culture, and then the subculture. The cycle starts, growing of the cells take place, then the secondary culture, then the subculture, and here we have the cell lines where we copy the cells, and we isolate the successive subculture from the turn, or if we are going toward the cytology research, so we do the single cell isolation. If we are like this we can say that towards many lines we can go here. So this is the place where we can divert. So dear student, this was the basic cell culture lecture. So inshallah, next lecture we will so dear students, this was the introductory lecture about the cell culture, in which we have studied what is the cell culture. We have studied about the history, we have studied about the scope, we have studied that what is going on nowadays and in the cell culture laboratories. Also we have studied that what we have achieved in the era of the cell culture, when we are talking that this is the era of the biomedical sciences, or we can sometimes claim that this is the era of the cell culture research and technology. So in the next lecture we will study about the cell culture procedures, about the cytotoxicity, about the cell viability, how we will determine the cytotoxicity and the cell viability. So this is all for the today. Thank you very much. So nice of you.